Okay, great. Thank you for being here. Welcome to the second to last HPS series that we found this year as at and today we have Caterina Schürch. I don't know if I said that right. So thank you for being here. So yeah, Caterina is going to talk about a problem that I think is very classic in discussions about how to integrate or not integrate history and philosophy of science, which is the accusation I think that most historians say of philosophers, right? That they are cherry picking and I think here presented in a much more positive light. Think about how we should do or we could do cherry picking. So the floor is yours. Thanks.
Thank you so much, Max and Assad, for inviting me. It has been very productive for me to come up with ideas for this talk. They could not all be implemented in what I'm going to show you. It is not as polished as I wished it to be, but I'm super happy about being here. I'm very thankful for you joining in. I'm super glad to present not only some research that was my PhD research, but also my thoughts about how I work. And it's been good to take a step back and to think about that for a moment again. After I did it, I think more when I did still study the history of science and history of and philosophy of science, then there was a more active engagement with these topics. And then I somehow ended up, after doing a Bachelor of Science in History and Philosophy of Science, I ended up in a history department, still being a philosophic-minded historian of science. And so today I'm speaking to you as a historian, being appointed as a historian of science at the Technische Universität in Berlin, but of course still interested in these philosophical issues. I'm super glad for talking to you about that. And cherry picking was the metaphor that I wanted to start with and maybe hopefully come back to in the end. My idea is that I'm going to talk about doing HPS, as you probably want me to, given the title of the colloquium, and then talk about the challenges that come with it, but mostly present you my approach, how I did it and want to do it in the future, the opportunities I see, and the plan for the talk is as follows. Need to enlarge it a bit. To think about why do we integrate disciplines and how? What is my particular approach? What can be said about that, apart from my particular approach about doing HPS in general, and what can be new avenues for me myself, but also maybe for the field and community of HPS. I try to do a parallel narrative of integrating biology and chemistry, which was the topic of my PhD. So I analyzed how biologists and chemists came to work together, but also I tried to use this now as a springboard for you to talk about how to integrate history and philosophy. So it will be maybe a bit an annoying switching between the actual work and the meta. I just want to warn you in advance. Maybe it's a bad idea to do it like that, but I myself found it fun when preparing it. Just to give a bit of a background motivation for what is good to come, you probably know the Society of Integrated HPS and their mission statement. It is looking as follows, and I want to highlight a couple of points. The claim that one can effectively advance history of science and philosophy of science simultaneously. It's one of the core assumptions of HBS and something that has been disputed and denied often. The claim that there is a common goal, which is understanding science, and then, of course, the big question is, what is the science that we are talking about? What is this exactly? Is it something really abstract that maybe has never taken place in history, or is it needs it to be something that has been done and then maybe not fulfills all the criteria that we wanted science to be? So there's a bit of a tension there. And I think much of the debate about HBS resolves about what is the explanandum, what are we really interested in understanding. And then the idea that we have to do good history of science and good philosophy of science when we do HBS. So we still have to adhere to all the standards of the disciplines. And this has been one entry point for the criticism to say it's impossible to, when we do history, then we leave whatever philosophers want us to do, and vice versa. Because as a historian, you need to ground everything you say in sources. You have to be really sensitive to the context. It's always local. It's always local in time and space. It's always particular. And as a philosopher, you want to be clear and simple and abstract and maybe universal in your statements. So that has been identified as a tension. So just let's keep that in the back of the mind, and maybe what I say during the talk can then be used to come back to this, these claims, and will help me to feed my take on that. Back to doing HBS and its challenges. One thing I want to suggest is that, something I decided for me to do at some point is not to care so much about all the things that could go wrong when doing HBS and to really concentrate on all the risks that we take, but more focus on if we want to do it, how can we do it in a good way? How can we even think about what does the discipline need or the group of people need in order to do it in a good way? So rather look for the opportunities and not so much just destroy ourselves by only focusing on the challenges, because it's not maybe a coincidence that HBS scholars have been so willing to engage in these debates about their own method, but maybe it, for my taste, it was, we were stuck a bit in these debates. Whereas it would be more interesting to think about the constructive side. So the big question for me is, how can we use our historical and philosophical resources and skills most effectively? And if we want to combine them, how does philosophy help me as a historian to do my work? And how can what I do help philosophers? And what is we and what shall we do? But let's get started. And now I leave this methodological discourse and I will present you with a case study. And we go back to the question why integrating disciplines and why, in this case that I looked at, have people in the area between the wars in the 1920s and 1930s decided to work on problems that needed chemistry and philosophy and biology to be solved. And the answer I found is that they did it in order to solve their problems. And in one of the cases that I'm going to start with, it was a problem in physiology, in sensory physiology, that was solved by using skills and resources from physical chemistry. The person who did that was Selig Hecht, who studied physics and chemistry in his undergraduate training, and then he went on to do a PhD in zoology at Harvard. And his whole career built on experiments that he performed in the year of 1917 in La Jolla at the Scripps Marine Biological Institution. And he worked with Siona intestinalis, that's an invertebrate animal, but sessile in one of its life stages. And the important thing about Siona intestinalis for Hecht's project was that these animals are sensitive to light. They would not be able to differentiate between different colors, but if confronted with a sudden illumination, sudden beam of light, they would retract their siphons. So just pull them back. And Hecht used, I told you, things from physical chemistry, especially the part called photochemistry, in order to solve the problem of what it is that happens in the sensory cells of these animals before the nerve impulse is triggered. And looking back to his career, he commented here then in these two books, one by Shepard, published in 1914, and one by Fitzweigert, a German photochemist in 1911, was knowledge which could be drawn on for an understanding of the nature of vision and photoreception. So the idea is we have knowledge from another field, and we can use it to achieve the goal that we want to achieve, which is a goal from another discipline here, physiology. And I worked out in my thesis how exactly this knowledge was helpful, and it was in terms of guiding Hecht how to design his experiments, from how to manipulate his study object to what to measure and how to interpret his data. For all these parts, photochemistry was instrumental. And I'll just give you a quick impression of this very first experiment, which went as follows. Hecht had 10 different animals that he collected the day before. The animals would be still alive, and they would be light would be applied to them, and the light would vary in its intensity. So we have 6 different intensities of light here. The way he varied intensity is just to, you would have the animal in this little dish below, have the Mazda lamp here, and then you would change the distance of the lamp to the animal, and this is how you would have a more intense beam of light at stage 6 and a less intense one at 1. So here we have these different values of intensity of light. 10 different animals. Every animal has been subjected to each intensity 3 times, and he would measure the time it took the animal to retract the siphon, because he found out in prior experiments that this reaction time was consistent for a given intensity and for an animal, more or less. So he had hopes for this to be an important data point. So how long does it take for the animal at different intensities to react to the light? And he found that the more intense the light was, the quicker the reaction was. And this, he knew, is one of the, it's the basic idea of the law of Bunsen and Roscoe. So we had, I'll come back to that. We see in his notebook that this is the law that he formulated in his notes. This is how he calculated these numbers here in the table. And there was photochemistry as a field that started in the 1860s, so way before it had to do with the chemical reactions on a photographic plate. So the photochemical effect that Bunsen and Roscoe were talking about was to the blackening of a photosensitive paper or and yeah, they formulated the law that it takes a fixed amount of energy to elicit a photochemical effect, and energy is the product of time and intensity, so you can either have stronger intensity and a shorter time of exposition, or a longer time of exposition and less intensity, but the product would be the same and you would get an effect. So Hecht found that his animals would show their behavior of a photographic plate in a sense. They would show a particular reaction, which is the retraction of the siphon, that obeys this law. And he argues that this means that the amount of energy required by Siona, the animal for a reaction, is the same for all intensities. This is the familiar phenomenon in the chemical effect of light. And that has some interesting consequences. He argues then, in terms of the processes that go on in the sense organs, the applicability of the reciprocity law means that during the sensitization period, the light must form a constant quantity of a substance before it can produce its stimulating effect. Idea being that something goes on in the sensory organs, we don't know what, but at the end of that process, there's a nerve stimulation. Apparently, it takes some time for this process to run, and the consequence he draws from the fact that the law of Bunsen and Roscoe applied to these animals and their reaction is that undoubtedly this involves the conversion of a photosensitive substance into something else, which acts then as an inner stimulus. Many such photosensitive substances are well known and have been studied with considerable care. So, from knowing that these animals react to light, but not knowing why and how, what happens on a chemical level, he found evidence for the thesis that there's a conversion that happens because of the chemical action of light on a photochemical substance into a non-photochemical substance. Maybe something else happens, but then the nerve trigger is instantiated. And then he went on, and a further set of experiments was designed in a way that he would expose the animals to short intervals of light after they were brought in the dark, and he would see that they would, a colloquial way of saying is that they adapt to the light, they get more sensitive. So whereas it takes a long time in the beginning for them to react, the longer they spend in the dark, the shorter is the reaction time. This dynamics of the reaction time, he said, can be translated in the fact that less and less energy is required every time, and that on a material basis, less and less of the substance P is required in order to get the effect. This, I don't know how mind-blowing it seems to you. It was, in many ways it was in the time, and in many ways it was not, because how was it not? In the sense that it was an idea that has been around for the longest time. As soon as photography was developed, there were hypotheses that this is how vision works. And as soon as photochemistry was developed in the 1860s, it was also clear to many photochemists and to many physiologists that probably this would be helpful to study vision. But But then nothing happened for a very long time. So everyone thought this is a really obvious idea, but no one was able to really prove it, to show it, to investigate it. Mostly because it was really hard to get to isolate the pigment responsible for light vision. And then Hecht's idea was to say, well, I don't need to isolate the substance, I only need to monitor its behavior and check whether it follows the behavior that we know so well from the photochemical substances that we have studied elsewhere in non-living systems. This is pretty cool, but it's even cooler. That's just something I wanted to mention. So, of course, you need a little bit of additional hypotheses for this experiment to work. You need to, for example, think that the nerve stimulation and muscle contraction, that takes next to no time, so that you can take the whole light reaction time as a proxy for the time used for light perception. Also, and we will see how important they are, he found these animals that he could use for the experiments. Animals that are slow enough in their reaction that you can really follow the time it took them to react. Animals that did not have any psychological apparatus, any optical apparatus that as vertebrates would have. Animals that would just allow access to studying the photoreceptive mechanism. But what is astonishing is that exactly at the same time, or here 4 years earlier, a physicist had the same idea, and he had the same idea of how to use the dynamics of the reaction to prove that it is a photochemical process, but he wasn't able to do any experiments because he did not have access to these experimental animals, or the idea that these particular animals could be used for that kind of experiment. There's another actor, it's a physiologist this time, August Pütter, in the same year as Hecht published the same idea, that sensory processes probably are these reversible chemical reactions, and he also provided an idea how to analyze that mathematically, also said that he would not be able to really do the maths, but he had help from his friend Gehenna Studi. And then there were people like Tokashu Nishika, who did this exact same experiments as Hecht did with the repeated applications of light beams. And they would just say, well, what we see here is an animal that gets used to light, and so it is quicker and quicker in responding. And Hecht would say about this explanation, it's not a good explanation because it gives us no insight into what really happens in the system. And these kinds of experiments about dark adaptation and how eyes get more sensitive the longer they are in the dark, they have been around for decades. That has been a really hot topic in physiology throughout. And the way these data were presented is to give the tables of how much more sensitive the study object became. And Hecht criticizes that this sensitivity and Empirigkeit says nothing. It just gives us the minimum intensity that is necessary to elicit a reaction, but do not know more about what happens in the system. And rather, he would interpret it in the way we saw, saying that, well, we need less of a particular substance to get the reaction. And this is what in Hecht's case, photochemistry, this knowledge, so sorry, that was a bit of a long excursion, but this knowledge really brought so it was a key ingredient for him to know, this is how I should design my experiment, this is how I can use my experiment to come up with a model of the mechanism of photoreception. And just to give you an impression of how extensive that input was, I have here in these titles, just the titles of the little book by Weigert of what he discusses, and we can see that it was a direct blueprint for Hecht of what to study in his experiments in the years that followed. So he had papers on every of these relations that Weigert discusses for photochemical reactions in vitro. So we can just go through this, and one of the important factors, for example, has been temperature. Hecht then in the next year used another experimental animal, which was this clam that I showed you before, that also has the same reaction of retracting the siphon here. And he would heat the water the clam was in with a little alcohol lamp, and then see whether in fact the part of the reaction time, that is the sensitization period, is running faster or stays the same depending on the temperature of the water, and he found it is invariant to temperature changes, which we know is the case for photochemical reactions. So another good hint that this could be a photochemical reaction going on. So, and with that, I want to step back again and say, could we maybe think about the relation of philosophy and history in a similar way that photochemistry was for Hecht and his project in physiology? In that, in my case, the literature on the structural mechanisms was important to design my work with the case studies. So everything that I would read about the new mechanism literature gave me hints of what I needed to look for in my historical cases. And in a way similar to Hecht, of course I did not perform any experiments, but it would give me an idea of how to choose the historical episodes that I'm going to focus on, what really to do with them, and give me an idea of how I could interpret what I found. And let me spell this out a bit, and just at the same time also say for a moment that we can, but for me it is, I can formulate the goal of the project in purely historical terms. So what I want to understand is why biological chemical research projects were initiated in the 1920s and 30s. And that question alone does not necessarily imply that I need the philosophy about mechanisms to answer that question. But why? And now it's the cherry picking in the other way around. So why would I, as a historian, pick the cherry of the new mechanism philosophy and not maybe something else? And what it made appealing for me was the fact that from, maybe you're all too familiar with that, so I don't really need to say anything about it, but...
No. I think
yeah, okay.
Don't be shy.
So it starts off with a characterization of what a mechanism is, namely entities and activities organized in a way that the phenomenon we're interested in is produced. And then what made it appealing to me is that from there were claims about research practice. For example, about what is the goal of biological research? What is to find mechanisms or to discover mechanisms? And for that, you need to find entities and activities and attach activities to entities and vice versa, and find out about the organization and think about what is the start point and the end point, and so on. So it gives us ideas about biologists really want to achieve. It gives us standards that need to be fulfilled. I need ideally a full description of how one entity and its activities lead to the other, so that you get from the start to the end point. And this is exactly what is needed to explain the phenomenon of interest and so on. People writing about mechanisms have then really described certain strategies of how biologists would proceed to get there. And they also argued that the search for mechanisms typically is an interdisciplinary endeavor. So normally you do not do it within one single discipline. One quote I have here is that the integration of biology is forged by building mechanism schemas that span many different levels, bridge across many different timescales, and that satisfy evidential constraints from many areas of biology, chemistry, and physics too. So in order to fulfill the goal that you want to achieve, you typically need the instruments, the skills, the resources from different disciplines. There is no particular, as far as I know, claim about how these collaborations come into existence, why a chemist and a physicist would be interested in cooperating with biologists. These are open questions, which for me as a HBS scholar is nice, because then I can make the point. Maybe there's something in history that could be informative and interesting for philosophy. That was the starting point. There's also a claim by many of these authors that many of them understand themselves as philosophers of science in practice, that what they describe is really informed by actual scientific practice and historical practice. And some of them said nothing in biology and the history of biology makes sense except in light of mechanisms. So this really the claim that this should help us understand the history of biology. So another, in a way, a clear signal for me, why not look for this, and a potential explanation for historical phenomena. So if I can really prove that they were trying to discover mechanisms in the way it is described in the philosophical literature, I can explain why they, for example, combined chemistry and biology. So I have like a blueprint for my argument provided. Before I show you how I operationalize this, it was important for me, and also vice versa, my colleagues in history, to establish that there really is need for this explanation, for an explanation of why people collaborated and mixed with biology and chemistry. Even though, of course, in history there have been ideas and theses about how cross-disciplinarity became a thing in the 1920s and 30s. They have argued, for example, we have just had the First World War, and especially in America, and how the war effort was organized. There now was a culture of collaboration. People were used to do collaborative projects. Now they have reasons to import that into science as well. Another explanation that has been given is that there was the Rockefeller Foundation, and it issued a program that was especially tailored to support biology that would use physics and chemistry. That was only done in 32. So a bit after the person we just looked at in the project, and Hecht even wrote to a friend of his, they're really intelligent to come up with that idea, but also they are travel slow in maturing, coming to the conclusion that this is good. But of course, that's often an argument brought by historians. We have to look at the financial structure, the incentives, the resources that are available for a certain kind of research. And then there was another line of explanation, which was that there was a tendency in biology at the time to distance oneself from descriptive biology, from morphology. So everything that was quantitative, that was experimental, was seen as good and as something that is progressive and something that brings you near to physics and chemistry and gives you the impression of biology being a science. But then, still, having all these explanations, which I do not reject at all, I argue that there is still not sufficient to understand what exactly has been done, because still, even if you are forced to work interdisciplinary as a biologist, for example, to cooperate with chemists, you have to come up with an idea of what to do exactly, and how you would combine your problems and your resources and skills. And I found some historical some evidence or some support for this idea. I have, for example, Friedrich Čapek, a French physiologist, who said, "It is well known how important and how difficult it is in physiology to select the right point of the complex of questions to be investigated, in which the lever of the physical method is to be used first." So you can be super okay with using physics and chemistry, but where and how exactly is not easy to decide. William Bayliss, a physiologist from the UK, said, although a student may have attended good courses in the physical sciences, she does not readily apply the knowledge to physiological problems, but it's because it's not readily apparent how. And looking from the other side, from biochemistry, Frederick Hopkins at some point said, the regular chemist has no aptitude for biological problems and does not recognize them. So it's really hard also if a chemist would think about what he could provide or she to biology, they would not know what the biologist's problems are. So finding a fit between the skills and resources and the problems of different disciplines, it's really, really hard. I would also say it's the main challenge for us as historians and philosophers of science. Should preface this. But how did I proceed in my own work? I had 4 different case studies and a similar set of questions for all of them. I started out by, for all these projects, to just describe what they did. And for that I used lab notes, their publications, letters they wrote, just the normal things you would use as a historian to establish that. Then I specifically worked out what they wanted to achieve, what kinds of methodological standards they were emphasizing, what their particular resources and skills were at the beginning of the interdisciplinary project. So what was, in a way, the starting conditions of these projects? Then the most specific point of analysis was what kind of ideas did the protagonists have about how things that were part of the different disciplines were connected. I can show you in a moment what I mean. But for example, for Hecht, it was the idea that these photochemical reactions are connected. The photochemical reactions are the things that connect photochemistry and physiology, because the photochemical reactions happen in an animal. Then the 4th section had to do with how then it really took place, how was the project implemented, what kind of strategies were used, what were the different steps, how did maybe goals and standards and resources used change, what kinds of alliances on an institutional and personal level came about, and so forth. And then in the end, how did the protagonists themselves and how did the people around them in the discipline evaluate the results of the projects? We already saw the example of Selig Hecht. and the action of the animals. I had another project another case study that was concerned with two groups, a group of botanists and a group of organic chemists in Utrecht. The goal here was to isolate the hormone that is responsible for plant growth. The botanists at the outset of the collaboration already have established that there must be a chemical substance. They did not how it looked like, what its characteristics was. And the chemist came newly to Utrecht, Fritz Kögel, and he was really into the business of natural substances chemistry, so finding hormones, finding vitamins, finding these kinds of substances. But he knew in order to find these substances, I need a biological system. I need these kinds of reactions in order to find it. And this is how they did it. So there was, they used little oat seedlings at the stage where it was still a coleoptile, so now the green leaf has not broken through. Touched the top end of these seedlings because it was clear that in the top end the substance was produced, and then apply candidate substances of the hormone onto the seedling, on just one half of it, and then see from the curvature how enriched the substance already is in the sample that they tried. So today we would call that a bioassay. The plant is just used as an indicator of whether you have the substance that you're interested in or not. And then there is a third case that I will explain later when I have a better slide for it. And then I had a fourth case that I won't tell you about anything at all, but it was a case that was not successful in the way that they didn't achieve anything that they themselves or their peers around would see as a breakthrough in the understanding of biological phenomena. With my remaining time, I would just give you very quick impressions of how I looked for these different things. For example, how did I try to identify the goal of Celic Hect? I would, at one point, resources like research applications were really, applications for funding were super helpful, because there you have to state your goal and you do it before the fact, which is, I found, more informative than the retrospective story they gave. But at this time, the applications were not as expansive as they are now. So for Hect, it was really informative also just to look at the publications and where he would formulate what is the goal of his whole project. And he gives this statement at one point, that finally to reach a more or less complete statement of the physicochemical nature of the substances and processes that underlie the photosensory responses of an animal like me, that is the goal of my work. And that, if we compare that to the expectation of the new mechanists, that is, I would say, that can be brought in that corresponds quite well. And then maybe the trickiest part was to find out about their methodological standards, because I didn't want to rely on their self-presentation in like more polemic pieces. So I also looked in the publications for the ways in which they specify what needed to be achieved in order for the problem to be solved in a satisfactory way. And here's one example when Hecht says an analysis of sensory simulation in order to be objective. So objective is a norm that he wants to fulfill, must take its data from the relations between the properties of a stimulating agent and those of the response of the animal. It also needs to be quantitative. So we need to be able to measure the simulation, but also the reaction in quantitative terms, and we need to, of course, be able to control it. And then he, in a German publication, said the end goal of the whole business is to understand the stimulation process in terms of the physical and chemical characteristics of the substances that are the components of the organ of concern. So we have these things in the publications, and then even more beautifully, I could trace that back to his own education. I found his lecture notes at Columbia University, where we see that, for example, he at least he noted the bibliographical details of an article by Beer, Bethe, and Uexküll, but also from his notes from the lecture of his professor, he really was taught that when we are thinking about sensory processes, we are thus required to know about possible environmental stimuli, exactly. And we also Parker, who gave the lecture, followed the suggestion of Bethe and Uexküll that we should define sensory processes in terms of the stimuli. So that's a really good hint that this is something that he learned to care for and then did care for. Another thing that he apparently cited in his book, in his lab book, was this article by a colleague of his who said, if we want to say something about the mechanism of visual response, we need to employ the definite structural and quantitative concepts of theoretical physics and chemistry, and then make dynamic arguments, and so on and so on. So this is how I learned more or tried to work out the protagonists' methodological standards. Then I needed to work out the resources and skills they deemed important, and for that, I could, for example, use things they bought and the receipts. Here is a postdoc from the group in Utrecht that then went to Caltech and founded his own group working on the similar project, and he immediately, again, ordered the same kinds of research organisms, these seeds of a particular line bred in Sweden, Swallow, or he built facilities where he could control the temperature and the humidity of the room, which was really important to control the environment for his experiments to work. So this is how you would learn about resources and skills. Also, another way, and this is how it then began to really click nicely with how I could combine these kinds of analysis with then things that we might want to explain with, which is why did here chemists and biologists need to collaborate in a project like this. We saw clearly that the chemist who came to Utrecht had an expectation that he needed biologists or physicians for his kind of work. And then on the other hand, let me give you again well, it doesn't really help. But then the botanists, on the other hand, they were super happy to help the chemists to find the hormone, because it was clear for them that the next step for their project is to have the hormone, to then know about its characteristics, but also to use it again in their own physiological experiments to see how exactly the hormones affects the plant. That again was important for the chemists to know, because they wanted to know, why is the hormone working? What are the characteristics of the hormone that make it biologically active? So this is a really nice way of how the different parties had different goals, but were interdependent in a way that it was clear that they could only achieve their relative goals together, and it was clear, because of this logical structure of the interdependence, that the botanists were ready to help with all the resources that they had. So that was 1 year of all of the manpower they had to just do these experiments in order to find the hormone. And then it was clear for Kögel, the chemist, to give them half of the substances that he found. So it really helps to understand also the social dynamics of the projects. And then I told you I was looking for the interfield theories, or what is called in the literature, like that is these ideas of how things studied in different disciplines are in fact connected. And here is now this other case study where a biochemist that was at the same time a geneticist in William Bateson's group, but also worked in Frederick Alan Hopkins' biochemistry lab, realized that for a particular set of pigments, the anthocyanins, these were pigments that in organic chemistry were isolated and characterized, and in genetics were the kinds of pigments that were responsible for the colors that you would trace chemically in your Mendelian experiments. So she realized here we have a very good opportunity to combine the two, to learn in fact about how exactly genes act and how exactly these pigments are produced in the plant. So she said the biochemical interpretation of factors or genetic factors for color is part of a greater problem, namely the origin of anthocyanin in the plant. There were two opposing ideas about that. I don't have to go into detail, but then just to give you an impression of the project, of how it developed, it was her PhD student, Rose Scott Moncrief, who was a biochemist. She was able to isolate these kinds of pigments from plants. Then there was the group of Bateson at the John Innes Center that was occupied with finding or with coming up with hypotheses of the genetic factors underlying these patterns of flowers. And then there was other groups, one in Oxford led by Robert Robinson, a very famous organic chemist at the time, that tried to synthesize these pigments. And in this project, Scott Moncrief had like a key skill, which was the ability to extract these pigments, because that was then helpful for both parties to advance their own research. For the chemists, it was really good to have a proof that what they have synthesized is in fact the natural occurring pigment. For the geneticists, it was really good to know what they have characterized as a phenotypic difference is in fact a phenotypic difference also on the chemical level, and that sometimes similar pigments would produce a similar shade in color, so that was a source of imprecision in the genetic project, so that could be helped with. But then, for these reasons, both parties were really happy to collaborate with Scott Moncrief. She, in fact, had another plan, following her teacher, William Onslow, in fact, with J.V.S. Haldane, to say, well, this is our key to understand what a gene actually does. So if we know in a specific case that we have between two plants only one genetic factor that is a difference, and we have from the pigments that we isolated out of these plants only one methylation factor that is a difference, we know that this genetic factor is responsible for this change in the chemical structure. So that was the breakthrough in chemical genetics, and explains again why from the beginning people were happy from both sides to collaborate with this third person, and this third person could pursue the project that she wanted to pursue, even though from the beginning not both of the other parties were really on board, because they were not expecting it to work, but it did in the end. But yeah, here again you see how a particular skill was a key for a decision for a certain collaboration to come into being. And then how did I assess how people evaluated these projects? We can see whether, for example, in the case of Kögel, his fellow chemists were happy with what he did. They gave him medals and awards and nominated him for the Nobel Prize. Wendt, the physiologist, also got nominated, and Kenneth Thiemann, another biologist, started out as a chemist. He got a kind of the Stephen Hales Prize. So when the field awarded them the prizes, that was a clear indication that they were happy. And if the protagonists themselves were happy, for example, here, Thiemann and Wendt wrote in their first book publication of the project that few fields can have benefited so much from the close interaction between the biological and chemical approaches, and the remarkable discoveries of the chemical side have made possible equally remarkable progress in physiology. This is a very quick run through what I did. Can I take 5 more minutes to then still enough oxygen in the room? So this is how, in the end, it was really, in my view, an effective use of philosophy, because many of the predictions made by the philosophers really were descriptions of things I found in history, and then would help me to, at the same time, explain why the chemist was happy to work with the biophysicist or the biochemist, but also why exactly a certain strategy was employed, or why at some point one of the parties lost interest. So it really made sense. I think I have new inputs for history of philosophy of biology about why the chemists in this case were interested in collaborating, because they had goals that they could not achieve without the biological systems and the skills of how to manipulate these systems. I could show that there were certain constellations of interdependencies between problems and resources that would reflect on how the research was organized socially. So we have, for example, this case where Selig Hecht alone was able to integrate everything because he had his background knowledge in physics and chemistry, because the photochemical work was already done independently, and he had an idea of how to combine the two, but he didn't need to collaborate with anyone. Then we have this case of a coordinator, the biochemist, who had offerings for both parties, who needed input from both parties, and then brought together these inputs in her own work. And we have this case of a collaboration on equal terms, where both parties from the outset were really clear about that they needed each other, and that they could not pursue the goals that were really central to their own discipline without the skills and expertise of and the resources from the other project. So that was really nice and neat. And that, so maybe here's the point where I can talk about the cherry picking on the other way around. So once I began to, of course that was not planned in advance and you cannot really do a historical work, but once it became clear to me that these are the structures that I will be able to show, it was also clear that maybe now these are good cases to compare to each other, and that Also with the fourth case of the failure, that I could make the points that I want to make on a more general level on the basis of these three case studies, because they were different in interesting ways. So it gave me, in a way, an idea of now I'm done, I have done my homework. Similarly, with another point that I will come to in a minute, but on the other way around, this set of things I needed to do that was also informed by the philosophy similarly helped me to structure my work on the historical case. It gave me some clear indications of what I need to worry about and what not, and that I need not go into much details about, for example, the religious belief systems of my protagonists, because I would, insofar as they would have affected their goals and standards and resources and skills, it would have been relevant, but as long as I checked these things out of my list, I would have everything I need to make the argument. It also gave me, in a way, a clear direction and focus of what to do. And this is, to come back to what I said in the beginning, how I think HDS can help, each other's discipline can help us to be effective and to know what to do and maybe what not to care about too much. I already said, what can I offer to the philosophers? On the one hand, an idea of why chemists and physicists would be interested in searching for mechanisms or being part of a research endeavor that is looking for mechanisms. On the other hand, maybe it's super apparent to you, for me it really helps to realize one thing, which is that the way the philosophers formulate their expectations about research practice is top-down or from the end result back to the beginning, in a way that they first characterize the end goal of the endeavor, which is the mechanism model, and from that develop what needs to be done, which is to develop, test, and revise a model, and what kind of skills and resources are needed. So this is thinking from the product back to how we get there. Whereas if I want to tell the story as a historian, it's important to start with resources and skills, or maybe formulate it in another way. It is not for any of these cases that people decided, oh, I want to learn about vision, or oh, I want to learn about plant growth. It was not curiosity-driven decision to work on a particular mechanism, but it was a pragmatic decision. It was built on the idea of what do I have, what are my skills, what are my resources. This Silikate already did his PhD work on Assisians. The lab of Fritz Wendt already worked on these coleoptiles for 10 years. Similarly, the projects on the genetics of flower color went on for 20 years by then, and the organic chemistry. So these are all established sets of skills and resources that have a history, and it's starting from these grounds that people thought about the problems that they decided to attack next. So now we can think about what is that a problem for philosophy? Does not need to be. It can just be the different things we're interested in. Of course, it's not the problem of the philosopher to explain why this particular person in that point of time decided to do this and that next. Still, it was important for me to realize that maybe that's still something that is a result of logical positivism and our fixation with just sentences, that we are interested in the formulation of the model, and that is how we analyze research practice. And we're not really taking the stance of the actor. But it could be just an input to think about that, if that's fine for us as philosophers to do it that way, or what it would mean to develop a philosophy of science in practice that takes the other stance. I think that's enough for today. I would have other things, but this is how I want to close. I think, yeah. So, yeah.
Okay. Yeah.
Water.
Yeah, let's have 5 minute break. Yes,
You
sorry.
have cold water.
Yes. Okay, nice. Thank you for this talk. Yeah, questions. I have some questions, but I want to open the floor.
Okay.
Yeah.
You said that you use as the new mechanism, you use entities and activities. So therefore, it's my camera about the engraver.
I'm closer to Bechtel, but
Yeah, that's
yeah.
my question. Because
Yeah.
why did you choose this one of mechanism? Because activities is not always obvious in your case.
Yes.
In your cases study, that activity is the right way to understand the mechanism.
Well, for example,
But just on the other hand, I can understand MDC. These people, they wrote much more about the standard, the strategy, the nesting of a mechanism, other tools that you use in other part of your work. So why choose this one?
There I was pretty pragmatic. I would not see how the difference between operations and activities would be relevant for my level of analysis. Activities were really important in the case of Hecht, for example, because he did not get a grip on the substance for a long time, but could only tell about the characterizations of the activities. How long does it take to have a photochemical conversion? How long does a normal chemical conversion reaction take place? But I just, maybe I was too pragmatic there, but I just took the two definitions as being as overlapping for everything I needed, and then decomposition and functional decomposition, that's a thing that Bechtel really focuses on.
They are pretty close, but
that was important here.
My impression is that the activities and entities works particularly well on neurons and neuron mechanisms.
Yeah.
Less clear in genetic mechanism or in biochemical mechanism. In that case, maybe operation, relation of parts, interaction, are often concept that seems better, but depends on the cases, of course.
Yeah. Yes, I mean, I'm open to, that could be a next problem to attack, to say, are there subtleties between these two takes that would that I could say something productive about by looking at these cases? Yes, it didn't strike me as really relevant to problematize. Yeah,
Okay.
yeah. But I'm of course open to that. I took entities and activities and components and their operations as more or less synonymous. And then also, of course,
on the other hand, if you ask Darden, clearly they say it has nothing to do with operation. It's activities what's chosen for a very specific reason.
Okay.
And Craver too would say that.
Yeah. Well, yeah. The chemists and the physiologists in the hormone case, they were talking about activity enabling properties.
Interesting.
Activity structure, that was the relationship that they talked about. And I would have to go into another presentation to really get the words used by these people. Yes.
But it's okay. It was fitting better your cases.
I just eclectically took from both strands of the but I'm open to discuss that more or to take more care into looking what the difference could imply.
I have a follow-up regarding that, which is, so I don't know so much about this literature on new mechanism, but so this notions of activities and entities, for you, they work both as actors category-ish and analytic category, sort of, is that those concepts are doing the work of bridging between your historical work and your philosophical interpretation in a way.
So in a way, everything that is mentioned here these are all connecting points during the bridging. So is it the way people talk about the goal and the norms? Is it about the way people talk about what they do and what they need to do it? These are all connecting points. And then I was, of course, not looking for the term activities and entities. But if Hecht would say something like I showed, the substances and processes, that was fine for me to say, here I have something like entities and activities.
Almost, it is also the structure of the sentence already feels very close to the...
It is, for Hecht, it's laughable how close it comes. Also when he talks about what his measurements are in relation to the target mechanism in the animal and then the interpretation. So yeah, you could really, if you would have to invent a cherry to suppose that it could not be done better. Yeah.
Cool. Thank you so much for your talk. Is another following up. I don't want to stick on this point, but maybe it could be interesting because another critiques for entities and activities is made by an article by Krieger and Marie Kaiser, 2018, I think I remember, in which they criticize, for example, activities. Not all biological processes fit necessarily the etymology or the meaning that you can give to activity. Or for example, there are some processes in biology that are passive, osmosis, etc. So I don't know if in your specific case, it could be interesting to check if in all your biological processes that you are interested in, or maybe not, it's a general, it is not for your case specific or necessary, it's a general, no remarks, general observation.
Yes.
Because they had a lot of problem in biology concerning the entities and activities.
But it's still a mechanism. So the main point is you switch to mechanism. So that's the important point in philosophy of biology.
Yes, that's still
among the mechanism you disagree.
But the meaning they provide some more general, less constrained notions. I don't remember which one, but maybe it's not. Sorry to.
No.
It's not. It's really only because I talked about this.
Yes, So I didn't come across. So I checked other. Of course, not only these 4 cases for whether I want to work on them, and I didn't come across others that did not have any activities in it. But on the other hand, I would not argue that there are not these cases. Something I can tell from this group that didn't achieve anything in the end is that in my analysis, the problem was exactly that they didn't have an interfield theory in terms of either entities or activities, but so whereas it was always clear, so for this project it was clear on the outside, photo perception has somehow something to do with chemicals and photosensitive chemicals. For them, it was clear that hormone action and growth, so the hormone, there's something about the hormone, and that was this lock and key metaphor that was really widespread at the time. Probably something about the structure of the hormone that is important for the way it acts. So they were really perplexed when they found out, in fact, that differently structured chemicals would have the same physiological action. That was a big problem at some point, but then they found a way around it. For them, it was clear whatever genes do, they probably do something on a genetic level. And it was established that, it was clear that also somehow these pigments in the plant must be developed somehow. So for all these projects, it was somehow clear what they were suspecting to be the relation. And for this project, it was more about, well, we assume that things that happen in biology has something to do with electricity. And they had no clear idea of what kind of phenomena they were interested in, what kind of behavior of the system, and what kind of electrical process on the physical-chemical level that they could really then pursue and test and make plausible. So that was just the lack of any ideas of what kind of behavior and activities they were concerned with, even though they knew it must be something that has to do with electricity and the charge of the particles, was the reason why they didn't arrive anywhere. So I found I could not say I'm positive. Every biochemical research collaboration of the time had the goal of identifying some particular activities involved in a biological mechanism. But for the ones that I looked at and that worked, it was an important part.
Thanks.
Yes. Thank you very much for your talk. I was curious about the fourth case, the one you didn't present. I was curious whether you can tell us something more about that, but mostly for its possible relevance concerning how it fits in your perspective on cherry picking.
Thank you. 2 words about the project. So it was located in Prague. It was paid for by a publisher that was not a scientist, a hobby scientist, who was convinced that electricity is all we need to know to understand biology, who was selling half of the newspapers in Bohemia. So was very rich, could hire a biologist, Josef Eckelhorn, to work for him, and he had already collaborated with a physicist called Reinhard Fürth. Reinhard Fürth was really happy to help out biologists. He would be happy to make his instruments available to them to help them develop some techniques to identify the electric charge of dyes. And these dyes would have been used to then livingly dye these Daphnia organisms and highlight certain structures. And then they thought, well, if we know the electric charge of the dye, we can then conclude the electric charge of the physiological structure. And that tells us something about the function of the structure. That was the main idea. And they were super happy that they were able to highlight certain functionally already recognizably complete structures with the dyes. And then they just did not get to the next step of what they now learn about the fact that this part was highlighted. Also, it was always, in a way, it was too complicated because they would also know the exact, it was many factors figuring in to make this argument a proper argument, and then they did not have a theoretical idea of what it would mean. And why it was important for me to have this case
Yeah, so
Yeah.
what kind of lesson can we draw from
Yeah.
this failure in the context of your perspective on cherry picking?
Oh yeah. So yeah. So my point that I in the end didn't make explicit here is that it is super hard to pick cherries in a good way. It's an art, and for me, these cases were cherries to pick because they were different in interesting ways and similar in interesting ways. So they are, all of them are instances in when people try to bring together the resources and skills from physics or chemistry and biology. Some of them were done as a one-man show, others have been done in groups. So this case gives me the idea that you can happily collaborate as long as you have maybe some starting idea of what you are after. But it will get tricky to really interpret, to come up with an experimental design and to interpret if you don't have an interfield theory, if you don't have an idea of what you're after, an entity or an activity that connects to a certain biological phenomenon. So you don't have an entry point. So in a way, it made, I think, my argument stronger to say these were, for all of these cases, the interfield theories, for the successful ones, the interfield theories have been formulated in advance, have been around for a long time. So the conceptual linking was not the most difficult part. The most difficult part was really to operationalize it and to find an organism on which you could then do the experiment. And to really have all the techniques, also the synthesizing techniques, the isolating techniques, the microchemistry that was needed in order to isolate the chemicals, to get a foot into the mechanism. And it did not happen here, and it could not happen because there was no idea of what the interfield relation would be, other than the really sketchy and concrete one. So, yeah, it was important for me to have a case where I find part of what I want to explain, part of the explanandum, which was for a long time a happy collaboration, but then one that did not produce any results. And then for me, the ability to be able to explain it was reassuring.
Thank you.
Maybe it's a related question, but a sort of for me it's very clear how the philosophical framework of new mechanisms, although I don't understand it, but it's clearly that guided your historical research, not just the choice of the case studies, but also the choice mostly right, because if I understand correctly, new mechanism is really about understanding biological sciences and neuroscience, and sort of this those case studies also fit those. But my question is, whether your case studies sort of push back in any way the theoretical framework, sort of saying, well, this either because it doesn't fit completely, or sort of ways in which. I don't know how to say it.
Yeah.
But whether the historical case study sort of brought something new to the philosophical framework that was not there, right? Because at the beginning, you started with this idea that both disciplines can thrive together in sort of the ideal HPS is that those thrive together. So this is a very clear case of how philosophy can make history thrive, but maybe I didn't get it. How is history making philosophy thrive?
Yeah.
Is it sort of saying, well, look at this case study, this doesn't work?
Yeah.
Like, for example, this case that you talk about homeostasis and being rather a passive, so it cannot be considered an activity.
So one of it is an indication about the explicability of the concept, which is when you look in the new mechanism literature, they say, well, biology is about finding mechanisms, and then if someone tries to date it, they would say, at least since the second part of the 20th century, and then I can say, well, look here, 1920s and 30s, it was really a natural thing to think about in these terms about your goals and your methods and what you need to do to achieve it. That's, I think, just a good information for philosophers to have, that their concept can be applied to these parts of biological practice and history. Probably not all of it, but there has been work along these lines in the past, first. Second, an answer to, well, if we assume that we need all kinds of skills and resources, in order to get there, we need either have people who can use these resources and have the skills, or we need to be able to bring them on board and being interested in biological mechanisms. And that is not something that new mechanism explains, why chemists and physicists should be interested in that. And I can give an answer to that goes along the lines of, well, it's in a way a methodological answer. These chemists needed knowledge about the behavior of the overall system in order to identify the entities, often vital activities they were interested in. And they also, when they wanted to theorize about how certain substances were synthesized in the organism, or how they act in the organism, what the characteristics, these are all important theoretical questions in chemistry. They needed the biological system and the biologists to find these answers. Second point, and then the third point was really, for me, maybe it's trivial and maybe it's not a concern, but still just to realize how much we in philosophy think from the end result back on the research process and not the other way around. Yeah. And it makes sense. I mean, you have a goal, you do what you think you need to get there. But in reality, if you would ask Darden why was Hecht studying the mechanism of vision, she would say he was interested in explaining vision. And I would say, well, he was interested in doing good biology. He thought hard about what he can do and what he can do over the couple of weeks that he has in the summers when he goes to Woods Hole or La Jolla. And so that was a constraint. So it was clear he needed to do something with marine organisms. He knew about physical chemistry, he had mathematical skills, he knew no one else in my group of peers can do that. It was a safe and also a safe way to be original, and a good use of the specific skills he had. So I think he would have been similarly interested in smelling as well. But the reason why he chose to work on the mechanism of photoreception has all to do with his contingent history, of his study background and his resources. And that's, it's not a curiosity-driven project, what is my range of actions that I can do, pragmatic, opportunity-driven project. And so that would be my remark also for the HBS community. So let's talk about what we as historians and philosophers can do, what our skills is, what the contingent material is. It's really contingent that I have the lab notes of Hecht and not of someone else. But this is why I chose him as a cherry, because it's a nice cherry that makes that a good case. Nothing else. But I think with my work, I can give you reasons why I chose this particular philosophical concept and the particular historical cases. And it was not an easy decision, somehow it was, but finding the cases and then really connecting everything, it wasn't easy. So I said, well, I did, of course, cherry pick, but it was work. And it's not something that is bad, but I would, my wish for us would be to say, well, let's pick cherries and pick the good ones. And really get into discussions on what is it that we now work on, what are the problems that are interesting in philosophy of science, what are the problems in the resources we have in history, and then find out how it could be.
You had exceptional archive. You have even these notes at university.
Yeah.
This is exceptional.
Yeah.
It's rare that we have so much
Exactly.
information.
And it's easier in that time than it is in the 18th century where I work on now, or it would be in reading recent history, because then everyone, well, if you have access to your emails, it's fine. But I would have letters, I would have his lab notes, I would have his applications. It was really fun.
That already also sort of guides the kind of questions, the kind of philosophical framework that is relevant.
Yeah.
Because if, like, for example, I have some case studies where I have to do a lot of work to reconstruct what they were doing, to even try to understand their theoretical concepts. So I have much more stuff about their connection to political parties. So it's much more easy to go to a philosophical framework that talks more about that and about the practices, because I just don't have access to that.
Yeah. Also, I mean, there's a new mechanical philosophy, there's another philosophical project which is to understand collaboration, which is to understand interdisciplinarity. Ustani would say we need empirical cases, we need historical cases to develop our philosophy of interdisciplinarity. So I think this is something I have to offer. But it's never, I think, maybe not with philosophical and historical work per se, but it's also, especially not with HBS, it's never the end of a problem solution endeavor. It's just but maybe the next iteration of talking about it. But to come back to what the feedback is to philosophy, I think it's these 3 points. Range of application, explanation how this is the social implementation in a way of the project, and whether we really want to reconstruct the research practice retrospectively, and just to be, just to know about that, that we do it.
Okay, yeah.
Yes, I have a couple of questions regarding more the details of the cases, specifically about this Hecht work. So you mentioned that when he was choosing the model organism, one of the concerns was that it would be something simple without, too complex neurophysiology, etc., etc., etc. And then you mentioned that you also worked on the plant photosensitivity, but was there already some preceding work on plant photosensitivity, and whether it was was it important to work on animals, or it was just his lab which was doing animal physiology, so they were continuing doing animal physiology?
Thank you so much. Yes, the applicability of the law of Banzhaf-Roscoe to organisms was first shown in plants in 1910 by a person in Vienna, and also this lab in Utrecht. So that was clear, and it was one of the poster achievements for the group of general physiologists, because then Jacques Loeb, who worked on invertebrates, could also show it for Hydra, that they also have photochemical reactions. Jacques Loeb, of course, was really important for the whole political, disciplinary politics, to push for quantitative experimental biology in the US, and it was a great mentor for Hecht. It was in that time, these people had also a bit the people in Utrecht working, but also Loeb, they would say, it's even the things we're after are probably the same in plants and animals. So we are interested in these processes on a very general level. So also that makes it important to work on really simple animals, because then we have the most direct access to these fundamental processes. And it was for Hecht, of course, I think he was drawn to these photochemical systems because he wanted to associate himself with Loeb. Importantly, he went one step further than Loeb, whereas Loeb would just tell, hey, look how great I found a case where a physical-chemical law applies to a biological process. Hecht made use of that finding and said, well, it tells me something about the biological system because I think it tells me that there are certain substances that are part of that system. In a way, he filled that finding with meaning. And he would be able to do so because he knew way more about physics and chemistry than Loeb did.
But was he directly student of Loeb? Or?
No.
Okay.
No, he was a student of Parker, and he only met Loeb after he did these experiments in La Jolla. Loeb published the journal. Loeb edited the journal for general physiology, which was the platform where Hecht published his first experiment here, and it was the second issue of the journal being founded. And Loeb edited it together with Osterhout, and that was the methodological impetus for having this journal, is to say we need to promote this kind of research that is quantitative and experimental and takes into account physics and chemistry. So it was a really nice fit. And then Loeb became a mentor of Hecht's. Hecht had a position in Omaha, Nebraska, and was very unhappy there because he had no physicists and chemists to talk things over, he had no access to libraries and so on. And then Loeb made sure that he would get a fellowship by the General Education Board, and then was able to go to the zoological station in Naples, for example. He also went to Cambridge. He wanted to go to work with Otto Warburg, with Emil Warburg first, and also with Niels Bohr. Some of the plans that he realized are not. But and eventually, in like 10 years later, he got a position as a biophysicist at Columbia University. And Thomas St. Morgan, who was also in the team with Loeb and also had to edit these books, the experimental biology monographs, he was the one that brought, who made sure that there is a position for Hecht at Columbia. So again, a lot of social, institutional things happening that has to do with methodological norms and whether people are able to fulfill them.
Marco?
Can I make another point? Concerning I'm going back to the contribution that Max mentioned concerning history, contribution of history from philosophy. I think you mentioned, if I understood correctly, that a contribution from philosophy say, look at these case studies in which they already taught in a mechanistic way. Now, this is, I think, a contribution that you mentioned. Is correct. But maybe we can push a bit this. I would like to know what you think about it, because unless the new mechanism, the foundation was by the article by McKenna, Gardner, and Craver in 2000.
Or you can also say Vincent 74. We can also say Kaufman even earlier, but yeah.
No, because you use, in your presentation, you use entities and activities by, so.
Yeah.
So, and
in my oral presentation, I didn't show it explicitly on the, but yeah.
Okay. No, because for me it's interesting because the context in which they propose the mechanistic explanation and definition of mechanism was at the molecular level using the neurotransmitter release. They were very into molecular biology. But in your case, you are kind of before molecular biology, kind of 10 years before, if we're talking about 2030. So could be even more interesting to say it's kind of, we have a mechanism thinking before new mechanistic philosophy, because no, what do you think? Because it's before, so entities, activities are referred to molecular stuff, to molecular mechanisms or ion channel, calcium, etc. But you're talking about some thought before all this molecular biology description. So, what do you think about if it's relevant? I don't know, because it's true that we have to be we are to agree about the foundation of the new mechanism philosophy. So maybe this is, but if we think about rather.
So I think that's maybe the cycle that I mentioned. Okay, these philosophical concepts came from a study of the practice of a particular practice, which was for Darwin and Craver, of course, neurobiology and molecular biology, protein synthesis, and so on. Macamar works also on Galilei, right?
Macamar what?
He is also early modern.
Yes.
Well,
yes.
he would.
He's a Descartes scholar at the
Yeah.
beginning, before switching to philosophy. of
So he
biology.
would not be confined to molecular biology, probably. He would also be happy to defend that there was mechanistic thinking in the 18th century and before. Bechtel used work on vitamins in the 1930s, also when he developed his framework. So I think from the beginning, it was not only neurophysiology and protein synthesis that was the material that has been studied in order to develop it. But of course, I would say the philosophers of science who are thinking about scientific practice, they always also have a particular field in mind, or are best versed in a particular field, and then maybe what they come up with is influenced by the particularities of that particular field, and then it's all the more interesting to take such a concept and take it into other contexts and see whether they apply. And then historically, many of the things that the people did that I studied then would then become what people later called molecular biology. It is a bit of a retrospective misconception that molecular biology was always about genetics and about molecular genetics. So these vitamin and hormone studies and how they act, that would also be counted as a molecular biology in the early 1940s, for example. So maybe it even helps us to readjust a bit our idea of, or it just shows the prehistory of molecular biology and how natural it was for these people to, I would say, to think about activities and entities or components and operations. But of course, that's one way why I think, and I could put it in my way, the people who will do HBS are probably, in my view, the people who are interested in research practice. And that could be the philosophers, and that part of philosophy who really want to understand what is going on in science. And that could be either by them assessing science as it is now and maybe using sociological methods or just their impression, or they look at historical episodes. And this is the meeting ground. And so there's, in my view, always this necessary cycle of developing something by studying a particular field or a particular phenomenon in science, abstracting from it and come up with a general philosophical idea, and then take it as we take models in the sciences and try to apply them to somewhere else and see what happens. And maybe there's interesting feedback again for the model.
Yeah. I have one more question.
I have a follow-up on this.
No.
Yeah, it's just a short follow-up on this topic. So you mentioned, but maybe I didn't just understand it, that there is this sort of historiographical narrative linking the origin of molecular biology to the activity of the Rockefeller Institute and Rockefeller Foundation.
Yeah.
All this work is before that, right? Or is it at least independent from it?
Everything started before the Rockefeller Society.
Yeah, okay.
started to, but some of them, of course, got money, and also the Rockefeller Foundation already paid in the 20s for this general education, bought fellowships to send people.
And was it already this sort of physical-chemical biology? Yeah, okay.
Yeah, already some line of work that they would have to fund. Yeah, but I would not support the view that this is like the beginning point of people thinking about how that they should integrate physics and chemistry into biology. I think there was an independent need felt by the actors themselves, independent of the funding structure.
Well, okay, I have one, but it's kind of a beer question.
Yeah.
So we like do it. Then if you don't want to answer, then we.
Smile the other way.
But so it's out. It's sort of related, but it's outside the talk. But it's something I'm struggling with, and which is at the beginning you start with the sort of potential HPS tensions and sort of benefits. And then the last one is about the disciplinary standards, that it's hard to adhere to both, and you see specifically the sort of local contextual thingy, grounded sources and so on, versus the clear and simple, and that's always a struggle, especially when you have to present in 15 or 20 minutes how much detail you want to go on and so on. Another maybe obvious disciplinary standard that is difficult to bridge because everyone gets mad is the normative component.
Yes.
So historians don't do that. They don't like doing that.
Yeah.
They don't like people doing that. And philosophers. Well, we had discussion about whether sort of the normative of philosophy is normative or whether it's sort of essential to discipline or not. People have ideas in this room about that. But for me, at least, it is why I don't study philosophy, is the normative component. Not just doing the evaluation, but at least thinking about sort of, okay, what are the normative components in here? What are the norms guiding research and so on. So I don't know if there's some normative component in your work or not. And how do you deal with that? If at all?
Yeah, sure. Thank you. Let me preface this by saying, of course, there's a reason why I'm interested in science and philosophy of science and history of science. And that has to do with, but then, for the most time, I can just say normative standards for me are interesting insofar as they have been important for the historical actors. And that I would say no historian can take from me, that occasionally when people think about what they do, they think about how the thing that they want to achieve needs to be achieved. Certainly when you are a postdoc starting your new project, you want to do good work, you want to make yourself a name, you have to really think hard about how to use the little resources that you have effectively. So this is why I think for every historical description of what happened, normative ideas of how you should do your work are always a necessary component. Now, a historian can say, well, still, for that I don't need philosophers to tell me what these norms are. And for me, it's more a thing that has to do, that is similar to this relation of Hecht and the photochemists. For me, it was easy to say, well, there has been people that have been thinking hard for a couple of decades about how the goal of finding mechanisms was achieved and how that, what the logical connections of that are to research practice. So why not just take their work and test them in a way for my cases? So let me rely on the experts who are really trained in finding the consequences of particular characterizations. And so for me as a historian, necessarily I need to talk about norms. I can always say it's not me who decides whether that was good science. It's also not my first interest to say, oh, these Prague people were bad scientists. But I do want to understand why, in their view, and the view of their peers, they were not successful. Or the other ones were so happy to say, we have achieved so much within so little time. That is something that I think historians should be able to explain, and I think they need to refer to the norms of their study objects. And then, for me, philosophy is a good resource to inform myself about norms. Always with the option that this would not help me to understand the historical events. But maybe it does. And if it does not, it also is maybe an interesting learning of philosophy of science.
Okay, yeah. Okay, great, thanks. Okay, and I think we close.